Hello everyone and welcome to my presentation. I am HOUSSAT, PhD student from Faculty of Sciences and Technology, University Mohammed bin Abdullah of Fez, Morocco. In this work I will discuss the application of the theoretical method called PEPSEA, peptidic search, to investigate the active structure of the chemotactic peptide and formyl-Methionine-Leucine-Phenylalanine. Polymorphonuclear neutrophils are cells that provide an effective defense against pathogens. Their chemotaxis can be stimulated by various informal peptides, such as FMLP, which are considered as structural analogues of bacterial products involved in the chemotactic response of neutrophil to infection areas. The tripeptide and formyl-Methionine-Leucine-Phenylalanine is therefore a powerful mediator to inflammatory reactions. It stimulates the release of polymorphonuclear neutrophils, superoxide O-2, and the lysozyme enzyme of the neutrophil. A systematic study of the relation of structure to activity has demonstrated a highly specific dependence of the activity of these peptides upon their structure. The specificity of the formyl peptide receptor located on the surface of neutrophil cells has been established. In the context of peptidomimetic, several modifications have been made, such as substituting the native residues by other bulky groups. The aim of these modifications is to limit the degrees of freedom of the molecule and then capture the bioactive tridimensional conformation of the native peptide. The influence of terminal groups has been studied, and it has been demonstrated that the substitution of the formyl group at N-terminal by the tertiobutylcarbonyl carbonyl group results in a total loss of activity. The substitution of the central residue with aminocyclohexane carboxylic acid and aminocyclopentan carboxylic acid resulted in an increase in biological activity. Using a theoretical method, in better to understand the role of three-dimensional structure in biological activity, our comparative conformational study of the parent peptide with these letters have revealed that the native conformational of this kind of chemotactic peptide is the beta-chain structure and the development of the pseudocycle C10. So we all try to introduce changes at the backbone by replacing the amide bonds by thioamide ones, which are more resistant to enzymatic hydrolysis. This replacement at the methionine level was carried out and resulted in a dramatic loss of activity. The comparative conformational study of the two molecules FMLP and FMSLP showed that the active chemotactic peptide must have the formyl group free of any intramolecular interaction in order to be available for the formation of the complex with the receptor. We present through this work a comparative conformational study of the parent active peptide FMLP and its thioamide analog FMLSP devoid of any activity. The main objective is to explain the difference in activity between the two tripeptides, to propose a native conformation of MLP analogs, and to determine the structural requirements for molecular recognition by their receptors. The method used in this study is called PEPSEA, peptidic search. It was developed by Mitchell et al. In the structural chemistry laboratory of the Sherbrooke University, and it has shown its effectiveness in the theoretical conformational studies of several peptide molecules and their structure-activity relationship. This approach is based on the fact that the structural, thermodynamic, and statistical properties of a molecular system can be deduced only from a population present in its conformational space. The principle of PEPSEA consists of generating a population of conformations that characterize a particular peptidic sequence. Then, a minimization of energy to the closest minimum is performed. Afterward, a statistical analysis is applied to this population of minima to deduce the thermodynamic and structural properties of the peptide study. The force field used by the PEPSEA program to compute the conformational energy is ESEP2, Empirical Calculation Energy Program for Peptide. This force field uses the rigid geometry to represent the amino acid residues of the polypeptidic chain. The conformational energy function is the sum of four terms: electrostatic term, Leonard-Jones term, hydrogen bound term, and the torsion term. The PEPSEA program described above carried out the conformational search and the localization of the most stable conformers. For each two considered tripeptides, 20,000 conformers were randomly generated and energy minimized to the closest minimum. During this generation, all torsion angles are allowed to vary, except the amide bonds, omega methionine, omega-leucine, and omega-phenylalanine, which are fixed at 180 degrees due to their double bond nature. We have carefully ensured that the 20,000 generated conformations subjected to minimization are the same for both molecules. For each tripeptide, the first 100 conformers of lower conformational energies were submitted to a second energy minimization, allowing all dihedral angles to be modified. The Hessian matrix was calculated, and the free energy was evaluated for each of the 100 minima. The resulting conformers were sorted by increasing values of the free energies, and then clustered into groups of conformers having the same structural characteristics: beta turns, gamma turns, or extended. The conformational analysis of the most stable conformers of the parent peptide FMLP shows that it can adopt varied conformational structures, with the majority can be distributed into four classes. The first class is represented by 43 conformers adopting a beta-turn structure centered on methionine and leucine. Figure inside gives the superposition view of the conformers belonging to this class, which are stabilized by a hydrogen bond between CO formyl and in each of phenylalanine. The second class includes 31 conformers characterized by conformations adopting a gamma-turn structure centered on leucine and stabilized by an intramolecular hydrogen bond involving the CO of methionine and NH of phenylalanine. The third class is that of conformers are characterized by the presence of another gamma-turn structure centered this time on the methionine residue and represented by 80 conformers. Such structure is stabilized by an intramolecular hydrogen bond involving the CO formyl and NH of leucine. The fourth class gathers structures in a double gamma turn, a gamma turn centered on methionine and a gamma turn centered on leucine at the same time, and includes 8 conformers. The determination of the native structure of the FMLP analogs can be deduced by comparing the conformational preferences of the two tripeptides. The fact that we found almost the same number of conformers adopting the beta-turn structure for the active peptide FMLP 43 times and for its inactive analog FMLSP 45 times rejects the proposition that this structure may be the native structure of chemotactic peptides. Indeed, if this structure was native, the thioamide analog would also have been active. For the same reason, the double gamma turn structure will be also rejected. The FMLP adopts this structure 8 times and the FMLSP, devoid of any activity, adopts it 10 times. The gamma turn structure centered on methionine third class is more preferred by the inactive thioamide analog 14 times compared to the parent peptide 80 times. Consequently, its change to be native becomes weak. The gamma turn structure centered on leucine is adopted by FMLP 31 times and just 5 times for the analog FMLSP. The gamma turn structure, centered on leucine second class, seems to be the native structure of MLP analogs. Firstly, because it is more preferred by the active parent peptide FMLP than for its inactive analog FMLSP. Secondly, it confirms the importance of the availability of the CO formyl and NH-leucine that has been mentioned in the literature. The comparative conformational study of FMLP and its analog FMLSP, described in present work, allowed us to deduce three fundamental aspects concerning the structural requirements of FMLP analogs receptor. The gamma turn structure centered on the central residue seems to be the native structure of the chemotactic peptide analogs. The CO formyl and NH at position 2 must be free of any intramolecular hydrogen bond and available for a constructive interaction with the receptor. The inactive analog FMLSP prefers a gamma turn structure centered on the methionine residue. And thank you for your attention.